Hello, and thank you for joining us for this live webinar, Characterization and Affinity Maturation of TCRmimic scFvs identified using Phage display. I am Sambhav Dave from AppCam, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by AppCam. As an innovator in reagents and tools, AppCam's purpose is to serve life science researchers globally to achieve their mission faster. Providing the research and clinical communities with tools and scientific support, we offer highly validated biological binders and assays to address important targets in critically biological pathways. To learn more, visit www.appcam.com. So let's get started. You can ask questions to the speakers during the presentation while they are fresh in your mind. To do so, simply type them in the Q&A box, which will open when you click the green Q&A button at the lower left-hand side of the presentation window. The questions will be answered after both presentations. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience any technical problems seeing or hearing the presentation, just click on the support button at the top right-hand side of the presentation window, or report your problem by clicking on the green Q&A button at the lower left-hand side. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page, and we will follow the process of obtaining your credits. I now present today's speaker, Jackie Douglas from Johns Hopkins School of Medicine. Jackie completed her bachelor's degree in chemical biological engineering and economics and a master's degree in chemical engineering practice at MIT. She is currently an MD-PhD candidate at Johns Hopkins School of Medicine and is doing her PhD thesis in the lab of Bert Vogelstein. Jackie's thesis involves using Phage display to identify TCRmimic scFvs, or antibody fragments, recognizing mutant peptide-HLA complexes, and translating these scFvs into therapeutic formats. Jackie will now begin her presentation.
Hi, Sambhav. Thanks so much for your introduction. So my name is Jackie Douglas, and I'm a graduate student at Johns Hopkins University in the lab of Bert Vogelstein. And today I'll be talking to you guys about characterization and affinity maturation of TCRmimic scFvs identified using Phage display. So first, an outline of my talk. Today I'll first provide an overview of what TCRmimic scFvs are and how they might be useful for cancer therapy. And then I'll provide an overview of Phage display and the panning process, which is how we go about identifying our scFvs. And then finally, I'll provide a case study for one antigen of interest. It's a KRAS G12V mutant peptide folded into HLA-A2. And specifically, I'll talk about how we identified an scFv recognizing this antigen and how we went about characterizing that scFv and affinity maturing it. So we are interested in targeting something called antigen processing and presentation. All nucleated cells in the body express class I HLA, or human leukocyte antigen, and this can more generally be referred to in other species as MHC, or a major histocompatibility complex. And so when proteins in the cell are degraded by the proteasome, a fraction of the resulting peptides will be taken up into the endoplasmic reticulum via the TAP transporter, where they're loaded onto HLA molecules and sent to the cell surface. These peptide-HLA complexes serve as a way for the immune system, specifically T cells, to sample the contents of that cell and determine if that peptide is a self-peptide or a foreign peptide. So during normal T cell development, the majority of T cells that recognize self-peptides are removed in the thymus, and so only peptides, the vast majority of T cells in the periphery are going to recognize foreign peptides. So I mentioned this is a mechanism for the immune system to sample the contents of cells. And so, for example, if a cell is infected with a virus, some of the peptides on the cell surface will be viral peptides, and certain T cells that have a T cell receptor or TCR that recognize this foreign peptide will be triggered to kill that virally infected cell, thus clearing the viral infection. So there's three class 1 HLA genes, A, B, and C. And these genes are polymorphic, but some alleles are more common than others. So for example, HLA-A2 is found in around 40 to 50% of the general population. And HLA-A1, A3, and B7 are also similarly very common. And so the reason that we're interested in this pathway for cancer therapy is that a fraction of intracellular mutations can be presented extracellularly, and this would be a truly tumor-specific marker present on the cancer cell surface. So now I've modified the diagram to contain the red star that represents a peptide-containing mutation. And so it's possible that peptides containing mutations will be presented as a mutant peptide-HLA complex on the tumor cell surface. So it turns out that many cancer patients, it's been shown in cancer patients that many of them have anti-cancer T cell responses, and that they have T cells with TCRs recognizing these mutant peptide-HLA complexes. And this can be protective against cancer. However, the tumor microenvironment tends to be very immunosuppressive. So drugs that are involved in checkpoint blockades, such as anti-CTLA-4 and anti-PD-1 antibodies, can help reverse the suppressive tumor microenvironment and take the brakes off the T cells to ramp up T cells that have an anti-cancer TCR. However, we know that for the vast majority of patients and the majority of cancer types, checkpoint blockade drugs are not enough. And so that's where the goal of our project comes in. Our goal is to create TCR-mimic antibodies targeting peptides containing common driver gene mutations presented in common HLA alleles. And that's because not all patients have a strong enough endogenous anti-cancer T cell response. We call our antibodies MANAbodies for mutation-associated neo-antigen antibodies. So importantly, our antibodies must be specific to the mutated peptide epitope and not its wild-type counterpart, and it also must not recognize any other normal peptide presented on a normal cell surface. And then once we identify an antibody, we can think about using it in a variety of different therapeutic formats. So this diagram shows a full-length IgG antibody. Full-length IgG antibodies work either via antibody-dependent cellular cytotoxicity called ADCC or complement-dependent cytotoxicity called CDC. And these are both fairly sensitive, but they're not necessarily the most sensitive therapeutic formats. And so we're also looking at converting our antibodies into CAR T cells, bispecific antibodies, and antibody drug conjugates. And so I mentioned that we're interested in targeting mutant oncogene peptides. And now I'll provide a little bit of background why specifically we're interested in mutant oncogene peptides. So cancer cells are driven by driver genes. Driver genes can be broadly broken down into two classes. We have oncogenes, which have a gain-of-function mutation, and tumor suppressors, which have a loss-of-function mutation. And because there's fewer ways to induce a gain-of-function in a protein than a loss-of-function, oncogenes tend to have stronger hotspot mutations. And one of the most important oncogenes that's mutated in cancer is KRAS. KRAS mutations are identified in around 130,000 new cases of cancer every year. These mutations are present in nearly all cases of pancreatic cancer and around 30% of colorectal cancers. And importantly for our purposes, mutations in codons 12 and 13 occur for the vast majority of KRAS-associated cancers. So this pie chart from the COSMIC database shows the KRAS mutation frequency. And you can see that the vast majority of KRAS mutations occur at codons 12 and 13, with G12D and G12V mutations being the most common. And a very small fraction of KRAS mutations occur at codon 61. So to identify our MANAbodies, we basically had two methods to choose from. There's two common methods that are used to generate antibodies. The most traditional method is called monoclonal hybridoma technology. And that's where you immunize a mouse with an antigen. And after a period of time, you're going to harvest the spleen and isolate the B cells to produce a hybridoma. And then what you do is you screen the hybridoma cells to look for the hybridomas that produce an antibody that binds to your antigen of interest. So there were a couple downsides of using hybridoma technology for our purposes. It's a slightly longer process. It takes around 6 to 9 months to generate a mirroring antibody. And importantly for our purposes, you cannot customize in vivo the selection process. And I'll explain exactly what I mean by that in the next few slides. So the second method to generate antibodies that's commonly used is what's called phage display technology. So in phage display technology, what you do is you take a whole library of different bacteriophage. So bacteriophage is just a virus that only infects bacteria. And this library of different phage, each phage presents on its surface a different antibody fragment. And what you can do through a process called panning is enrich for phage that display an antibody fragment with desired antigen binding properties. So in our case, we're using an antibody fragment referred to as a single chain variable fragment, or scFv. And that's where you directly link together the variable heavy region and the variable light region of an antibody as a single chain. And this is the minimal portion of the antibody that retains antigen binding. So phage display technology has several advantages. It only takes around 1 to 3 months to generate a human antibody. And if we want to use these antibodies as therapeutics, they need to be humanized. And then finally, for our purposes, we can customize the antibody selection process. And I'll explain what I mean by that in the coming slides. So our MANAbody platform consists of a number of steps. First, we have identified common cancer mutations and driver genes, particularly oncogenes. And then we predict which peptides containing those mutations will bind to common HLA alleles. So not all peptides will bind to a given HLA allele. Peptide binding is really dependent on the N and C terminal amino acids in that peptide. And so we use online algorithms to predict if a given peptide will bind to a given HLA allele. We've built a custom scFv phage library. And then we do a process called competitive panning to enrich for phage that present an scFv that binds to our mutant peptide HLA complex. And then finally, after we identify phage hits, we can sequence the scFvs and recombinantly express the scFvs. And we use those recombinantly expressed scFvs for downstream assays, such as ELISAs and cell-based assays. So now I'll go into a little bit more detail about how phage display works and what the panning process entails. So the diagram shown shows an M13 bacteriophage. This is one of the most commonly used type of phage for phage display. And what you'll notice is that this phage contains a phagemid DNA surrounded by a number of different viral coat proteins. And what I want to do is draw your attention to the P3 coat protein on the left side of the screen. The P3 coat protein is present at 5 copies on the phage particle surface, and this is typically used as a fusion partner for an antibody fragment, such as an scFv. So to build our phage library, we generate a library of variant scFv sequences, and we subclone that library into what's called a phage midplasmid, such that the scFv DNA is just upstream of a P3 protein DNA. And then what we do is we transform this phage midplasmid into E. coli, and then we infect that E. coli with helper phage. So the phage midplasmid provides the P3 coat protein, but you have to co-infect the E. coli with helper phage to provide all the other viral coat proteins as well as the DNA packaging machinery. And thus we can produce our full phage library. So to use our phage library, we do a process called panning. And panning, you take your phage library, you incubate it with an antigen of interest, you wash away any unbound phage, and you can elute any bound phage using acidic conditions. And then we typically amplify that eluted phage by infecting E. coli with our eluted phage, and we grow up phage overnight. And we can repeat this panning process multiple times to gradually enrich for a specific phage. Now this diagram shows positive selection, where we want phage that bind to a particular antigen, but you can also imagine doing negative selection, where you first incubate your phage library with an antigen that you do not want your phage to bind to remove any binders, any phage binders that would bind to that target. So our phage library, as I mentioned, is an scFv-based phage library, and the framework of the scFv is based on the trastuzumab Herceptin framework. This is also referred to as 45. The reason we chose this framework is it's very stable and well characterized, and it's been shown to work well for other previously published synthetic phage libraries. It's a fully human framework, and it's very similar to the canonical human IgG1 sequence. And importantly for our purposes, it allows for stable, high levels of recombinant protein expression. So at the bottom of the screen, shows a diagram of the fusion protein found in our phage midplasmid. So at the end terminus, or at the 5' end, is a PELB leader sequence, followed by the scFv sequence, and we have a MIC tag, a TEV cleavage site, and a full-length M13 P3 protein. So in designing our scFv phage library, we wanted to add diversity in the regions most important for determining antigen specificity. And typically these are referred to as the complementary determining regions, or CDRs. So this diagram shows a full-length antibody. There's 2 heavy chains in green and 2 light chains in blue. Each of the light chains and heavy chains has 3 CDRs represented by the red lines. So in antibodies that would be produced by the body endogenously, it's the CDRs that are the most hypervariable and thus determine antigen binding. So another advantage of using the 45 framework is that the crystal structure for the 45 scFv has been solved, and so we know which of the 6 CDRs is exposed to solvent, and thus is the most important for determining antigen binding. So of the 6 CDRs, L3, H1, H2, and H3 are considered the most important for antigen binding. And so it's these 4 CDRs that we chose to vary in our phage library. So this table shows the amino acid diversity in our library. In CDR L3, we only varied 4 positions by 2 amino acids. We introduced either a serine or a tyrosine in these 4 positions. However, in the other CDRs, we had increasing amino acid diversity. And additionally, you'll note in CDRH3, we also, there's a delta symbol. This represents a random deletion of that position. So at a certain probability, we deleted that position so that we had variable length diversity and CDRH3, as CDRH3 is considered to be the most important for antigen binding. So the DNA for our library is synthetic. It was generated using degenerate oligonucleotide design, and it codes for the select amino acids shown in the table. We outsourced the DNA synthesis for the library. However, we constructed the library in-house. And what I mean by this is we did the E. coli transformation of the phage plasmid in-house, followed by infection with helper phage. In designing our library, we wanted to avoid any stop codons or cysteines. I mentioned that we had variable CDRH3 length. And the final library size is approximately 5 times 10 to the 9th unique clones, which is on the high side for phage libraries. And then after building the library, we performed next generation sequencing to confirm that the library reflected the specified amino acid diversity and length diversity that we wanted. So now I've talked a lot about the phage panning process and our specific scFv phage library, but now I want to talk about the antigen that we use for panning to identify TCRmimic scFvs. So we use what's called a monomer. A monomer simply refers to a soluble HLA peptide complex. And a monomer consists of an MHC class 1 alpha chain, and this is actually just the ectodomain of the HLA molecule. And this is folded together with a peptide of interest. So for example, in our case, it's going to be a mutant peptide or a wild type peptide or any other control peptide that we want to investigate. And this is folded together with beta-2 microglobulin. And this final monomer complex can be biatenylated. So the monomer mimics the peptide-HLA complex on the cell surface. And the biatenylation is really useful for our purposes. It allows for easy in vitro manipulation, such as conjugation to streptavidin magnetic beads or streptavidin agarose, and we're going to use this conjugation for panning. Or for downstream assays, what we frequently do is we conjugate our monomer to streptavidin ELISA plates. So to go about identifying TCR-mimic scFvs, it's actually a very challenging process because we're looking for an scFv that can distinguish a single amino acid change in a complex that consists of around 480 amino acids. So to do this, we've developed a series of phage selection steps to slowly amplify mutant peptide-HLA specific phage. These steps consist of 3 phases. The first phase we call the enrichment phase. So at the beginning of round 1, we take library phage, and we perform negative selection by incubating our phage with streptavidin magnetic beads, as well as denatured monomer. So in every prep of monomer, there's going to be some denatured monomer, and so we want to remove any phage that would bind to the denatured HLA. And then we take non-binding phage and then perform positive selection, where we incubate the phage with a mutant peptide-HLA monomer conjugated to streptavidin magnetic beads. And after around an hour, we wash those beads and elute with acidic conditions, and we amplify that phage by infecting E. coli with the eluted phage as well as helper phage. And we grow up the E. coli overnight to produce more phage. And then we repeat this process typically around 2 more times. So that's the enrichment phase. The second phase we call the competitive phase. In this case, we're going to take round 3 phage at the beginning of round 4. We're still going to perform negative selection in a similar manner. However, in this case, we're going to take the non-binding phage and do what we call competitive panning. So with competitive panning, we co-incubate the phage with both mutant monomer conjugated to streptavidin magnetic beads and wild-type monomer conjugated to streptavidin agarose. And in this way, we can set up a competition situation where basically a phage that has higher binding to the mutant monomer, but may have some low level binding to the wild type monomer, can still be isolated. And so what we do is after about an hour, we use a magnet to pull out phage that are only binding to the mutant monomer, and we can wash these magnetic beads And similarly, we amplify that phage. And so for competitive panning, over the course of rounds 4 through 8, we typically drop down the ratio of the mutant monomer to the wild-type monomer, and thus increase the stringency of competition. The final phase is what we call final selection. And in this case, we're going to do negative selection against the wild-type monomer and other control monomers to finally get rid of any phage that would cross-react with either the wild-type peptide or other places on the HLA molecule. And then we take the non-binding phage and once again perform positive selection against the mutant monomer. And we typically repeat this process one more time. So what you'll notice about this process is that unlike traditional hybridoma technology, phage display really allows you to fine-tune the selection process. So with hybridoma technology, there really wouldn't be an easy way to do negative selection and positive selection in vivo. So now I'll provide a case study for one antigen of interest. This is a KRAS G12V peptide folded into HLA-A2. So I mentioned that KRAS G12V mutations are the second most common mutation in KRAS. And HLA-A2 is the most frequent HLA-A allele type in the vast majority of ethnic groups. So the peptide we're interested in is a 5 through 14 peptide. And you can see that the G12V mutant peptide and the wild-type peptide only vary by a single glycine to valine change. So following the panning process, we titer the phage by infecting E. coli with limiting dilutions of phage such that each E. coli will only be infected with a single phage particle. And then we pick single bacterial colonies and inoculate those bacterial colonies in a deep 96-well plate. We grow up that bacteria and infect the bacteria with helper phage, and thus we can produce monoclonal phage. So then we do what's called a monoclonal ELISA, where we pellet the bacteria in the deep 96-well plates, and then we look at how the phage-laden supernatant binds to either the mutant peptide-HLA complex or the wild-type peptide-HLA complex on an ELISA plate. And then finally, after we identify any hits on the monoclonal ELISA, what we do is we re-amplify hits for further characterization. So this bar graph represents one of the monoclonal ELISAs that we saw for the KRAS G12V HLA-A2 antigen. The y-axis is a readout of the ability of the scFv to bind to the protein of interest. The blue bars represent individual scFv binding to the mutant KRAS monomer, and the red bars indicate wild-type KRAS monomer binding. And what you'll notice is that for the majority of scFv clones here, they either exhibit no binding to either monomer or they exhibit very similar binding to both monomers. However, there was one clone, we call it D10 for the well position, that exhibited much higher binding to the mutant monomer than the wild-type monomer. So we re-amplified the D10 phage and characterized it again on ELISA to confirm that indeed this D10 clone was specific to the mutant monomer and did not recognize the wild-type monomer. So then we wanted to recombinantly express the D10 scFv for other assays. So we worked with Axiomx. Axiomx is a subsidiary of Abcam, and we worked with them to recombinantly express our scFv in bacteria. And they were able to add a C-terminal flag tag that we use for a number of downstream assays. So one of those assays was an ELISA. So this is an example of one of the ELISAs that we did with the D10 scFv. So once again, the y-axis just represents the scFv binding ability. We tested the scFv at 4 different concentrations represented by the different colors. And you can see that when there was no monomer coated on the strep plate, there was no signal. With the G12V HLA-A2 monomer, we saw a nice dilutional signal. For other KRAS HLA-A2 peptides, including the wild-type 5-14 peptide G12C and G12D, we didn't see any bindings. And for other KRAS peptides folded into HLA-A3, similarly, we did not see any binding, as well as for other all other monomers we tested, including EGFR peptides folded into HLA-A3 and ELA, which is just an HLA-A2 control peptide folded into A2. So this was really promising that our scFv seemed to be specific on ELISA. However, this is only using recombinant protein on a plastic surface. So we wanted to see if our scFv could actually recognize a cell surface peptide-HLA complex, as this is the ultimate goal if we're going to use it as a therapy. So to go about answering this question, we use a cell type called T2 cells. T2 cells are a lymphoblast suspension cell line that is deficient in TAP, meaning that they're very inefficient at presenting their own endogenous peptide, and you can add a peptide exogenously and they will present that peptide. And this is referred to as pulsing T2 cells with a peptide. And this is a really good way to determine the specificity of the scFv for various peptide-HLA complexes. So we did various T2 peptide pulsing experiments, including flow cytometry. So in this case, we peptide pulsed our T2 cells. We stained the T2 cells with the D10 scFv, and we followed that with an anti-flag PE secondary. So the y-axis, the diagrams below are just histograms, and the y-axis represents the cell count. The x-axis represents the ability of the scFv to bind to cells, with increasing binding to the right. And you can see with the first histogram on the left side, we're comparing KRAS wild-type peptide pulse T2 cells versus mutant peptide pulse T2 cells, both of which are stained with the D10 scFv. And you see nice separation between the two curves. Similarly, for all other HLA-A2 peptides we tested, there is no D10 scFv binding. So one of the other HLA-A2 peptides we tested, we call LLG, and we also didn't see any binding. And importantly, if you look at the histogram on the right, when you take mutant peptide pulse T2 cells and don't stain them with the scFv, you don't see any binding, or you still see that nice separation between mutant pulse cells and all other conditions. So we took our D10 scFv and we wanted to convert it to a full-length antibody. So to do this, we simply replaced the CDRs in the 45 antibody called trastuzumab with the CDRs in our D10 scFv to produce a fully human IgG1 antibody. And then we transfected the heavy plasmids with the heavy and light chains of our D10 antibody into HEC293 cells for recombinant expression. And then we wanted to test, as frequently with scFv grafting, sometimes the scFv does not retain the specificity as a full-length antibody. So we did various downstream assays to confirm that indeed our antibody retained the D10 specific binding. So in this experiment, it's a flow cytometry experiment where we peptide pulse T2 cells. And in this case, we stained with a D10 antibody followed by an anti-human PE secondary. And if you look at the histogram on the left, this is comparing wild-type peptide pulse T2 cells represented by the black dotted line versus mutant peptide pulse T2 cells represented by the solid red line. And you still see the nice separation between the mutant peptide pulse cells versus wild type. And similarly for other HLA-A2 peptides we tested, like ELA, this same separation remains. And then if you look at the histogram on the right side of the screen, you can see when the mutant peptide pulse T2 cells are stained with an isotype control antibody, you still see about the same level of separation between mutant peptide pulse cells stained with the D10 MANAbody versus all other controls. So this was really promising, but we wanted to see if we could affinity mature our D10 clone. So if we want to think about using the D10 scFv or full-length antibody as a therapeutic, we thought that we might need a higher affinity, given the fact that we don't think the antigen density on tumor cells is necessarily going to be very high. So we worked with Axiomx to affinity mature the D10 clone. We provided Axiomx with a D10 scFv sequence, and they generated a Phage library of a D10 scFv variants using their Axiomx error-prone PCR mutagenesis, and their final library size was around 10 to the 8th unique clones. And then Axiomx performed panning. They did 3 rounds, each consisting of negative selection against the wild-type monomer, followed by positive selection against the mutant monomer. At the end of panning, Axiomx screened 88 colonies on ELISA, all of which recognized the mutant monomer. And then they sequenced the top 16 clones that gave the highest signals, of which 8 were unique. So then Axiomx, to do follow-up characterization on these 8 unique clones, they recombinantly produced these scFvs and tested them on various ELISAs. So now I'll give you some examples of the ELISAs that Axiomx performed. So one ELISA was just to see how the parental D10 scFv and its 8 affinity matured variants bound to the wild-type monomer. So Axiomx is referring to the parental D10 scFv as AXM2894. In this ELISA, the wild-type monomer was coded at a high concentration, and each of the recombinant scFvs, either the parental D10 or one of the affinity matured variants, was incubated with the wild-type monomer. And you can see that, like the parental D10, none of the affinity matured variants had any wild-type monomer binding. And this is really impressive. So frequently with affinity maturation, you may see some increased liability for other antigens that you do not want your scFv to bind to. However, in this case, we were able to get 8 different affinity matured variants, all of which exhibited no wild-type monomer binding. And the next ELISA that Axiomx performed, they did a titration ELISA. In this case, they coded mutant monomers at different concentrations, as shown on the x-axis, and they incubated the mutant monomer in each well with either the parental D10 scFv, represented by the light green line, or one of the affinity matured variants. And you can see that the parental D10 scFv gives the lowest signal for each concentration of mutant monomer. All affinity matured variants gave a higher signal, representing higher affinity. So in summary, all 8 variants had improved mutant monomer peptide-HLA binding with no wild-type peptide-HLA binding. So then we wanted to see if we're going to use the D10 scFv as a therapeutic, it needs to be able to kill cells that present the peptide-HLA complex. So we did what's called a complement-dependent cytotoxicity assay. And this is where you basically mix an antibody containing an Fc that binds to a particular target cell. And you mix this antibody, the target cell, and complement. And the antibody will fix complement via its Fc region and kill that target cell. In this CDC assay, we were comparing the D10 scFv to one of the affinity matured variants that Axiomx identified. And we call this affinity matured variant D10-7. In this case, we were using V5 tagged scFvs. And what we did is we pre-conjugated the scFv to a full-length anti-V5 antibody. And that's because, as I mentioned, to carry out a CDC assay, you need that full-length antibody containing an Fc region to fix complement on the target cell surface. So this is an example of the results that we saw with our CDC assay. So I'll just walk you through the diagram. The x-axis represents different concentrations of the scFv antibody conjugates. The y-axis represents specific cell death. The blue line are unpulsed T2 cells incubated with various concentrations of the affinity matured D10-7 scFv conjugated to the anti-V5 antibody. And you can see that at all concentrations tested, we essentially see no cell death. Similarly, when you incubate the D10-7 affinity matured scFv with the wild type peptide pulse T2 cells, you see very minimal cell death. However, when you incubate the affinity matured D10-7 scFv conjugated to anti-V5 with the mutant peptide pulse T2 cells, you see nice dilutional killing of the T2 cells with an EC50 of around 0.8 nanomolar. And this is actually in sharp contrast to the parental D10 scFv, which only had an EC50 of around 11 nanomolar. So this demonstrates not only can we use our scFvs for a killing assay, but that the affinity matured variants perform much better than the original D10 and are far more sensitive. So in summary, we've designed and built an scFv-based phage library. We can isolate scFvs that specifically recognize mutant peptide-HLA complexes over their wild-type counterparts. We've identified scFvs at this point for a number of different mutant peptide-HLA complexes, including the KRAS G12E peptide and HLA-A2. We've been able to successfully convert our G12E-A2 clone D10 into a full-length human IgG1 antibody. As well as working with Axiomx, we were able to affinity mature that scFv into higher affinity variants. And then finally, what we're currently working on right now, and I didn't show any data on this today, but we're working on converting our scFvs into therapeutically useful formats. So I mentioned that we've converted our scFv into a full-length antibody, but this may not be the most sensitive killing method for a therapeutic. So we're working on converting the scFv into very potent killing formats, including CAR T cells, bispecific antibodies, and antibody-drug conjugates. And then finally, I want to just make a quick plug for Axiomx's services, because Axiomx's services really made this project possible. We worked with Axiomx on multiple aspects of the project. So Axiomx provides custom scFv expression services, where they provide affordable scFv preps with the tag that you desire on the scFv. They can affinity mature an scFv, and I mentioned, I talked about how they allowed us to do the panning process, but we've worked at this point with Axiomx on a number of different affinity maturation projects, and they've allowed us to customize how we want to do that phage panning process. And then finally, I didn't talk about this today, but we're currently working with Axiomx on bispecific scFv expression in both bacterial and mammalian systems, including diabodies and scFv-SCs. And then for labs that do not have their own phage library and do not want to build their own phage library, Axiomx provides custom discovery screening using their own Axiomx phage library. And then finally, I just want to relate that this was a team effort. This project was a team effort. I worked with a postdoc, Andrew Scora, and another graduate student, Michael Wong, on all aspects of the project. I also want to thank my PIs, Bert, Ken, Shabin, Nick, and Louise, as well as the service providers, AbCam and Axiomx, the Hopkins Kimmel Cancer Center Flow Cytometry Core, as well as the Fred Hutch Immune Monitoring Core. Fred Hutch provided all the HLA monomers that we used in this study. And then now I want to hand the presentation over to Sambab, who will talk more about the Axiomx phage display platform.
Thank you, Jackie, for sharing your project with us. Following on the theme from Jackie's presentation, I would like to tell you about AbCam's efforts to generate recombinant antibodies using Axiomx phage display platform. AbCam serves life science researchers globally from sites in North America, Europe, and Asia with innovative reagents and tools. AbCam provides the research and clinical communities with highly validated biological binders and assays to study important protein targets in critical biological pathways. So recombinant antibody reagents offer many advantages over traditional antibodies. Recombinant antibodies can be designed to meet your exact requirements. They can be engineered towards specific affinity and the right applications, including IHC flow cytometry, and Western blotting. They are highly versatile in their applications. They are routinely developed as humanized therapeutic agents, and scFvs can be incorporated into an IgG framework for either diagnostic or research use. They can be engineered into chimeric antigen receptors and also bispecific antibodies. Recombinant antibodies offer a high level of consistency across batches of the reagent, and new batches of the antibody can be produced very quickly when compared to traditional methods. When it comes to picking the right platform to discover antibodies, no single approach is suitable for all requirements. And at Abcam, we have the choice to use Abcam RabMab rabbit monoclonal platform or the Axiomx phage display platform, each offering distinct advantages. The RabMab antibodies, discussed in detail in our other webinars, offer high affinity antibody binders when compared to mouse monoclonals. The rabbit immune system can recognize diverse epitope, and the affinity maturation of the antibody occurs in vivo. The Axiomx phage display platform became available to Abcam following acquisition of Axiomx Inc. in November 2015. It is a rapid antibody discovery platform which uses phage display library to drive the discovery. The affinity maturation of the antibody occurs in vitro using Axiomx mutagenesis. And today, I'd like to tell you more about the Axiomx phage display platform. The platform consists of M13 scFv phagemid library, and the library contains more than 10 to the power of 10 clones. The library is highly diverse with less than 10% parental clones, and the mutations are focused in the CDR region. The platform offers rapid liquid screening, which is performed in antigen-coated wells. The in vitro affinity maturation is performed using Axiomx mutagenesis, which is 1,000 times more efficient when compared to error-prone PCR alone. It combines site-directed mutagenesis and error-prone PCR to create a library with 10 to the power of 8 clones, distinct clones. The platform can also use whole cell panning and screen for binders against membrane-bound antigens on the cell surface. This ensures that the binders are screened for desired extracellular epitope of the cell surface protein. The Axiomx platform have distinct advantages over traditional methods of antibody discovery. Apart from engineering binders against protein targets that include peptides and fragments, the platform is highly suited to generate binders against post-translationally modified proteins. It can also generate high-affinity binders to antigens that fail to induce an immune response, or against non-protein antigens where traditional methods fail to produce usable antibodies. The platform is uniquely able to generate binders against difficult antigens that can be toxic to an in vivo system. A key advantage of the phage display is that binders can be engineered for the desired applications. The hybridomas can be converted to recombinant antibodies, and properties of an existing antibody can also be further optimized. So the Axiomx platform is divided into two main phases. The first phase is the discovery phase, and the second is the affinity maturation phase. In the discovery phase of the scFv development, using the diverse phage library, the goal is to identify candidate scFv clones that bind to the target with the desired specificity. In the affinity maturation phase, the scFvs identified in the discovery phase is then further enhanced by improving its affinity multiple-fold. This is achieved using Axiomx mutagenesis and additional rounds of rapid liquid screening. The final scFv is then further characterized in the desired applications and can be converted to a full IgG antibody. The rapid liquid screening approach that is used in Axiomx platform uses antigen-coated wells. This eliminates the need for any solid phase biopanning. The phage library is incubated in multi-well plates coated with the antigen of choice, and the wells are then washed to remove any unbound phage. The bound phage is then eluted, and the eluted phages can be either subjected to further rounds of screening for higher specificity or selected as candidate clones for characterization. The affinity maturation helps enhance the affinity of the candidate binder. The Axiomx mutagenesis uses a set of mutagenic primers and combined with error-prone PCR create a phage library derived from the candidate clone. This newly generated library is again screened against the antigen to get the desired affinity. So when comparing Axiomx mutagenesis to error-prone PCR alone, the Axiomx mutagenesis offers greater diversity in the phage library and an increased rate of mutation. So affinity maturation can also help improve the properties of the identified binder. Here, we have shown previously how affinity maturation can improve the stability of the binder and also help enhance the affinity of a given binder. The graph on the right shows using affinity maturation, we were able to improve on the original clone and achieve a 300-fold increase in the affinity for the antigen. And the graph on the left-hand side shows how affinity maturation improved the thermal stability of another binder. The original binder lost nearly 85% of its activity when within 7.5 minutes at 70 degrees centigrade. Using affinity maturation, we enabled the binder to remain active for over 60 minutes at 70 degrees. So the techniques used in Axiomx platform has been published in several peer-reviewed journals. The link to the articles can be found in further reading section of this webinar. The articles go in detail about the techniques, including the whole cell panning process and the affinity maturation process. You can also learn about other techniques used in the Axiomx platform, including emulsion panning and scFv to IgG subcloning, which I did not get to talk about today. And to learn more about Abcam's efforts to develop recombinant antibodies, please visit this link below. So in summary, the Axiomx phage display platform is a versatile method for rapid phage display antibody development. It is fast and effective and applicable to broad uses in life science research. So using recombinant antibodies, Abcam continues to improve antibody-based assays. And we have developed a number of singleplex and multiplex assays using recombinant antibodies. With this, I will end my part of the presentation, and I would like to thank Jackie again for her presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window, and Jackie and I will answer as many questions as time permits. Okay. So the first question is, have you been able to identify specific scFvs to each mutant peptide-HLA complex you've panned against?
Yep, Simbab, so that's a really good question. We have at this point panned for around 20 to 30 unique peptide-HLA antigens, and I would say that we've been able to identify scFvs that are specific for about half of those antigens. And I think that's true for a number of different reasons. In some cases, the mutant amino acid might be buried in the HLA cleft, and so the mutant peptide and the wild type peptide are going to look very similar in the HLA complex. We've also noticed that it seems to be a little bit easier to identify scFvs that recognize hydrophobic amino acids. So what I would say in summary is that about half the time we can identify scFvs, and some antigens have proved to be far more difficult than others. So.
Great. So another question is, if a peptide is predicted to bind to a given HLA allele, what is the chance that it is actually presented?
Yep, so this is a hot area of tumor immunology right now. So we know that not all peptides that are predicted to bind to a given HLA allele will actually be presented. So there's other variables along the way. That peptide has to be properly processed by the proteasome and loaded onto the HLA complex. And so no, not all peptides will bind to a given HLA allele, or sorry, not all peptides predicted to bind will actually be presented on the cell surface. And so we and other groups are currently doing mass spec and other assays to try to confirm that these peptides are actually presented.
Okay, I have another question for you, Jackie. Given that the Phage library only introduces diversity in the CDR, how important is diversity in the CDRs versus in the framework region?
Yep, that's a really good question. So because our Phage library could only be of a particular size, we wanted to introduce diversity in the regions that we thought would be the most important, and this is the CDRs. However, what we've noticed with the affinity matured variants, so I should say that with affinity maturation, you can get mutations in both the CDRs and the framework region. And what we've noticed is that many of the affinity matured variants have framework mutations, and this seems to provide increased affinity.
Great. So there's another question. Can scFvs be converted to full IgGs? And I can answer that. Yes, the scFvs can be put into an IgG framework, and Axiomx has multiple vector framework options that can be used. The next question is, do scFvs work in IHC?
So I can answer that. So scFvs do work in immunohistochemistry. We've done this many times. I didn't show any of that data today, but all you have to do is incubate your cells with the scFv of interest and then follow that with the appropriate secondary antibody. So yes, they do work well in IHC.
And from AppCam's perspective, yes, we can test the clones to ensure that they are screened for IHC. So the next question is, how do you devise the Phage library of scFvs? I can answer that. And you can add to that. So at Axiomx, the scFv library is a human synthetic library prepared by combining gene sequences together with randomized CDRs and to produce the Phage library. Jackie, do you want to add onto how you've developed your library?
Yep. So just to confirm, so there's usually two ways that you can make a Phage library. The first is that you can take endogenous B cell receptors from people. You can sequence those, and you can make a Phage library from those sequences. The other way, which is the way that we did, is that we it's a purely synthetic DNA sequence. We're using the 45 framework, and we're using various degenerate oligonucleotide technology to vary the amino acids and the CDRs.
Great. So we have another question for you, Jackie. Could you describe your NGS strategy? Targeted or shotgun? And what's the read length? And the sequencing instrument used?
Yep. So we use a MiSeq. We designed primers that flanked CDRs H2 and H3. Our amplicon, I believe, was around 150 to 200 nucleotides. And so we were looking at diversity in the CDR H2 and H3 region.
Okay. What software did you use to analyze the NGS data for your original libraries?
So our lab does a lot of sequencing, and we have an in-house pipeline to analyze data. But some of the analysis I just did with Excel, but that was very downstream analysis.
Okay. Another question on affinity maturation. How do you make sure that the affinity maturation of the antibody undergoes all the steps that normally occur in a lymph nodes?
So the affinity maturation we're doing would be different than what happens endogenously. So there's a number of different physiological steps that occur in the lymph node. It's going to be a different process, but the result is still similar in that we're getting variation in the CDRs and in the framework region. I believe from my medical school classes that most of the diversity that would occur in an endogenous B-cell receptor is going to occur in the CDRs. I don't think you're going to see as many framework mutations. So I think it's a different process.
Okay. The other question is for me. So does Axiomx have access to use scFv library for discovery? Yes, we do have a selection of libraries that we can use for discovery of an scFv. And I think we have time for one last question, and that is for Jackie. I have a question regarding the selection of cancer-associated mutation. Is there any criteria, and how can we predict peptide-HLA binding?
Yep, so our goal is just to help the most number of patients possible. So our criteria, we look at two things. We look at the mutation frequency and the HLA allele frequency. So we've created a score to, basically we've predicted all peptides containing common mutations, and we predicted which of those peptides will bind to common HLA alleles. And we've created a score just in-house to rank various mutant peptide HLA complexes. So basically that's our criteria. Other things that we care about is any evidence in the literature to suggest that that particular mutant peptide is actually presented. This evidence could be mass spec evidence, or it could be T-cell based evidence.
Well, thank you, Jackie. So I'd like to thank the audience. So I'd like to thank the audience for joining us today and for their interesting questions. We'd like to thank LabRoots for organizing this, and also AppCam for underwriting today's educational webcast. Please note, a short survey has appeared for you to answer. We appreciate your feedback on this survey as we work to plan for future webinars. This webcast can be viewed on demand through June 2017, and LabRoots will alert you via email when it's available for replay. We encourage you to share the email with your colleagues who may have missed today's live event. So until next time, goodbye.